I'm your host, Dr. Jeff Brown, with your Emerge Medical Podcast with my co host, Dr. Jeff Cole. I'm very excited about the group that we've got to interview today. This is the executive team from DiscGenics. This is a clinical stage biopharma company developing an allogeneic injectable disc cell therapy for lumbar degenerative disc disease. We're joined by Dr. Kevin Foley, inventor of multiple patents that have revolutionized interventional spine care, including my own spine. Thank you, Dr. Foley. His devices are used globally to improve patients' lives. He's currently the chairman of the board at Sims Murphy's Clinic in Memphis, where he practices neurosurgery. We're also joined by Flag Flanagan. Flag is the CEO with over 30 years of experience in the medical device field. We're also joined by Bob Winelak, very skilled medical device and biologics executive with over 30 years in the orthopedic spine market. With that, I'm going to turn it over to Flag to give us a brief overview of their pitch deck, and then we're going to go through with some pointed questions.
Jeff, thank you so much for having us on today. We really are excited about where we are. As you know, DiscGenics is a clinical stage biopharma company developing allogeneic injectable disc cell therapy for lumbar degenerative disc disease in the mild to moderate area. We believe we have a target population in this country of 2.5 million people annually that we can treat with an injection in a treatment room of this cell therapy. The cell therapy has demonstrated a regenerative capacity and potential to treat underlying pathophysiology of DDD, both disc architecture and inflammation. We were granted a RMAT, Regenerative Medicine Advanced Therapy designation recently based on this 1-2 data, which Dr. Foley will discuss, and a fast track designation in 2019 based on the preclinical animal studies we did using human cells. We have a scalable, built for purpose allogeneic manufacturing facility based in Salt Lake City, Utah. We have 33 issued patents, 26 pending, with many trade secret protections around our manufacturing and our know-how. We have de-risked the clinical pathway so far, and we're looking forward to pivotal phase 3 trial expected to target pain and disability. This is our leadership, and 3 of us are on today. Dr. Najib Thomas is a well-recognized spine neurosurgeon based down in New Orleans, Louisiana at Southern Brain and Spine. He also co-founded Crescent City Surgery Center and Surgical Hospital, where he currently serves as medical director. Colin Novick is a interesting fella. He comes from Japan. Since he was a year old, he's natively fluent in Japanese. We worked with him as a consultant, and he was Dr. Sharona, who led our Series C round of funding. He was Dr. Sharono's choice to be the board director after the Series C round of funding. You can see that, Jeff, we've had a very good history in terms of basically building our value and hitting our milestones from the time we were founded in 2008 through translational science, preclinical studies, and then you can see the rounds of funding along the way with the US IND, Investigational New Drug Allowance, by the FDA, June of 2019, followed by a CTN, which is similar drug allowance by Japan, with this fast track designation, and then the last patient enrolled in the US study with another round of funding to build out the manufacturing facility, with the last patient being enrolled in Japan in April of 2021, where we then received positive US interim data, and then at 52 weeks, and then at 78 weeks, and then the full 2-year data with the FDA granting us this RMAT designation. In the US, we worked through the FDA, the biologic branch, following a biologic license application pathway. And these are for various biologic drugs similar to Humira, Tremfya, and other biologics. In Japan, we are following the pathway for regenerative medical products, and we really are hopeful there at the possibility of a conditional marketing approval, possibly by the end of this year. We are seeking to raise a total of 95 to 125 million in our Series D round of funding. We've raised 71 million to date. We're raising 40 to 50 million currently in a convertible note to that Series D. We have $30 million of existing demand from the insiders on the convertible note now. So we're trying to fill out the rest of that. We have a recent Big Four accounting firm valuation that is very, very good. It's into the low billions of dollar valuation today. So we're very excited about where we are there, and we believe that we can be a potential blockbuster drug by year 3 of commercialization. That would be with revenue in excess of $1 billion.
I'd love to run through really the main topics, and Jeff Brown and I will kind of tag team back and forth, try to keep us on task. And again, we can edit this down. So let's get going. Who would like to tackle just the main discussion of the problem itself? Low back pain and how much of an issue that is in the United States and abroad.
So I can speak to that, Jeff. Chronic low back pain is an incredibly common problem. There's an incidence lifetime of over 80%. Back pain is the second most common reason patients visit a physician. It's the primary cause of lost work days. And although there are a variety of causes for back pain, degenerative disc disease turns out to be the most common cause. We spend as a society over $100 billion a year in the US treating this entity, and we don't do a very good job of it. We do not have a treatment for degenerative disc disease, or DDD, that addresses the basic underlying pathology and leads to any biologic repair of those discs.
And can you talk about what type of, I know you said this is a very big problem, but if one of you could answer what you feel like the total market size would be, like what's your total addressable market, both US, domestically, and worldwide?
Sure. Kevin, I can take this one. So there's been a great paper published in 2017 by Ravindra et al., where they looked at the incidence of lumbar back pain. It affects 250 million patients around the world. In 16 million patients in North America are affected annually by an incidence of low back pain. And we've done a lot of metadata around the Ravindra paper and other papers, and we've really distilled down sort of that modified Furman 3 to 7 that Kevin mentioned to these various patients, where we had a modified Furman 4 was the average patient in our study, and we've identified 2.5 million patients annually that conceivably we could serve with an injection in a treatment room utilizing a C-arm of IDCT. And if you look at that patient population, Jeff, to give you some idea of the proportion, last year, 2.1 million patients were diagnosed with cancer in this country. So it's a very large population in which we believe we can serve. And we really do believe there is this evidence that we are regenerating the disc from the inside out, while seeing these dramatic reductions in pain, improvement in ODI, and increase in quality of life that's substantiated by the disc volume increases we saw.
That's fantastic.
Dr. Foley, what can you say about the current standard of care, and where you feel that this company is going to disrupt, and maybe put some other modalities in the sideline?
Sure. Patients who present acutely will always have a non-interventional, non-operative approach first, right? So we'll continue to use exercise programs and therapy and manipulation and various medications, Jeff, for patients who present with acute simple low back pain. It's when the problem persists and doesn't respond to the typical non-operative interventions that we need something better. And for those patients right now, their options are limited. They can have a fusion, which, as you know, basically involves getting rid of the disc, replacing it with bone, placing some kind of hardware in the spine to help that fusion heal. And then we can never undo that fusion. So for patients with degenerative disc disease, for example, I personally don't like to do fusions because I'm taking away the function of their disc and putting stress on the adjacent level, right? That we know over time that a certain number of those patients are going to have issues at the levels next door because of that intervention. And that's not to say that all fusion is bad. It isn't. When you have true instability of your spine, when the bones are moving vis-a-vis each other, fusion is the right thing to do. So I'm not saying fusion will go away. Fusion will stay with us. I just think that the fusions that are done for disc degeneration alone are going to diminish in number with a treatment that can directly affect the underlying biologic problem with those discs. Another alternative is to place an artificial disc in that disc, typically two metal pieces with some polymer that can move upon each other. Those have been around now for a couple of decades, and clinically in the neck, in the cervical spine, disc replacement has proven to be useful. In the low back, it's just not proven to be very popular amongst surgeons because the outcomes haven't been as good. Probably because a real disc isn't two pieces of metal with some polymer in between. It's a complex biological structure. So I believe that this intervention will displace some of the more aggressive surgeries that are done now to treat the patients who haven't responded to non-operative measures. I also think because of the durability we're seeing with this treatment, again, at least 2-year responses to a single shot, if not longer, I think this is going to turn out to be really, really cost effective for the whole system. Not only a good thing for patients, but a good thing for the healthcare system.
Excellent. Well, a lot of the viewers will know that I practiced interventional pain for a number of years, and it can be at times frustrating with what you have available to take care of patients because everything we have has side effects. You're limited as not being a surgeon, I'm saying you're limited to NSAIDs, Tylenol, opioids, which come with a whole host of problems that we're all very much aware of. But there's really nothing like this. Even spinal cord stimulators aren't as effective for axial back pain as we would like. Having said that, speak to your solution, and particularly patents, where you are in terms of FDA clearance and the science of what DiscGenics is bringing to the market.
So I'll go ahead and touch on that. So patents, multiple patent family, 26 issued. We have around 30 in play and multiple families. And Flag touched on this earlier here, but our initial product is for the lumbar indication, but our IP covers the approach as well as the methods and then the derivatives of use for this here. And we talked earlier about how scalable and how these cells can be differentiated into different therapeutic outputs in here. And then we further, expanded our IP to cover the process improvements for the commercial scale that Flag touched on earlier in there. So we built up this tremendous, IP portfolio. And then even if we handed someone a patent with all the trade secrets to make something like this as a GMP compliant product, you couldn't do it. There's so much technology and R&D that went into being able to produce this as a drug product with these types of therapeutic outputs. Took us years to do. So we're very proud of where we are with that. But once again, the novelty of the approach is really being able to produce a tissue-specific progenitor cell from disc tissue that gives it the unique properties to be able to go into the center of disc, put out extracellular matrix, and literally rebuild the disc from the inside out at the same time, giving out immunomodulatory signals to treat the short-term pain and inflammation going on, really gives the special properties to these cells.
It's very unique, Jeff, from any other product that's out there in terms of the cell itself. We do have existing IP out to 42. That's 2042. So we're in a good way. Obviously, we've done, almost 13 years worth of basic science, pre-clinical work, with 13 to 14 different animal studies along the way, which everybody else is going to have to do to follow on. Then they're going to have to do the clinical work in a phase 1, phase 2, and then a phase 3. Which again can be another 7 to 10 year pathway. We've seemed to advance it a little bit more here from an accelerated standpoint, we hope. So we feel like we've got a very good head start. The other competitor in the marketplace now for a low back pain cell therapy product is Mesoblast. They use a selected mesenchymal stem cell that they use around other parts of the body, where they've done some clinical trials around cardiovascular studies and graft versus host disease. They do not have an approval yet. And they did fail their first phase 3 trial in their low back pain study. They're going back for another one. So we feel like we're in a very, very good position, especially around the fast track and the RMAT to move forward. And with the data that we've shown so far in this, and one thing, Mesoblast did not show an advantage over the normal saline product, and it did not show any radiographic improvement like we did with disc volume improvement. So we feel like we're in a very good position. And, again, we're building a lot of moat along the way, which is, happens when you build out a biologic and take it down this very, very rigorous regulatory pathway. So we feel we're in a very good position to literally help millions of patients around the world.
That's fantastic.
Fantastic. You answered what was going to be my next question about competitors, but it certainly does sound like you're in an open lane.
We are. We've got some open field running, and we're white hot focused on this particular treatment and this particular patient population, and then we think we can grow it from there. And we really like to use Edwards Life Science as one of our models. They got into the cardiovascular surgery and thoracic surgeon space, and they developed products over time, and they brought in the cardiovascular interventionalists and was able to kind of coalesce, that whole group, very similar to neurospine, orthopedic spine, and interventional pain, where, they became, one of the most successful companies in the world, from both the ability to help cardiovascular therapies and cardiovascular disease states dramatically, along with being one of the better investments in the life science space over time. So we feel like we're in a similar position and really can do that. We've just got to get through with the IDCT first, and then continue to grow our business to really help a lot of people. And that's been Kevin's, Bob's, and my mission from the very beginning. And it really is about, if you take care of the patient and if you're doing the right thing by the patient, everything else tends to take care of itself. And we've seen that in various things along our careers. As you know, Kevin, he's considered by many the father of minimally invasive spine fusion. And you know, one of the things I've referred to with Kevin along the way was what we call not FDA rigor, but Foley rigor, where he was adamant in the very beginning that we use human cells in the animals. You know, originally with Dr. Kukikoff, he had used animal to animal, you know, the same. And Kevin was, once we saw and Kukikoff's concept of selecting, enriching, and expanding these cells from human disc tissue, he said, look, if this is really going to work and truly be allogeneic and immunoprivileged, then those cells should show immunoprivilege in the animals, which they did. So, you know, there were some, you know, Kevin's really been amazing in terms of his intellect and his knowledge and expertise as the chief medical officer of this company to drive this product from a scientific standpoint and a clinical standpoint. And, you know, Bob's done a phenomenal job as he had at Osteotech with the execution and the execution risk along the way. And, you know, I'm just very, very proud and honored to be associated with it, to continue to maintain the discipline so that we're white hot focused to get this first product to market.
I find it fascinating that these human cells do not provoke an immune response in animals.
Well, it goes to the true immunomodulatory markers of the cells. And Kevin, I'll let you expound on that from what you saw in you know, this whole xenographic notion.
Yeah. So, you know, Jeff, as part of that, the output of our culture process, those cell surface markers that are, you know, friend or foe, right? When a cell comes into a living creature, they're downregulated, and so the cells are just treated as friend.
How about business model and go-to-market strategy? It's exciting that you're coming out of the lab and we can talk about next steps. I'd love to hear.
Yeah. So the next step is this pivotal phase 3 trial that we'll do. And this is a much larger patient population. We're going to look at both single and multi-level opportunities to serve patients. And we anticipate from a commercial standpoint that we will set up a pharmaceutical drug model with a direct sales force. And we will also do a lot of direct to patient marketing around, you know, and educating the patient with lumbar disc disease about our product and how it might work. And then we originally or initially want to start out utilizing centers that really are integrated spine centers that handle the whole episode of care. And we think that's one of the ways that we can start this out right. So there's a very good discipline about patient selection in this modified Furman 3 to 7, and move this forward and expand it into other aspects of the spine, possibly around cervical, possibly around adjacent level disease, where a patient has been fused and he's 5 to 10 to 11, 12 years later. And I say he, my wife, had a 2-level cervical fusion, and then 13 years later had another fusion because she had adjacent level disease. So we think we can expand, you know, up and down the spine for various episodes where we can interdict along the way in the spine, but we also believe that we can expand with another cell around the musculoskeletal system and then possibly other cells in the future.
That's awesome.
We want to ensure that we're having good success, you know, with the sites and in the following the labeling there. Work very closely. We touched on payers earlier, working very closely with the payers to get into this treatment algorithm in there, and then working with, you know, different medical societies and patient advocacy groups that are looking at degenerative disc disease and lower back pain, and really have a lot of grassroots marketing built in there combined with the pharma type advertising going on, and really work to channel these patients into the treatment centers so the patients can get screened properly and you know, and injected and treated with the highest standards of care.
I hadn't thought about until Flag you brought it up, but adjacent segment failure is a huge problem after you fused a patient. I think it's something like 10% per year per segment fuse if you take all comers. But being able to use this on the adjacent segments, has there been any thought to prophylactically doing that?
Devin?
Oh, yeah, so it's a great thought, Jeff. You know, we're going to have to study that, right, to know. But does it make sense logically? Makes a lot of sense. Because if you look at those adjacent segments, one of the first things you see those discs that were healthy start to change, right? And it's from the biomechanical load, and so why not intervene early? And maybe we'll save those patients a future surgery.
Yeah, I
That's
mean,
profound. I mean, that's just profound,
Jeff, and it's close to home with my wife. I mean,
right?
And I mean, she would have done anything while she was in incredible hands for this adjacent level fusion.
Right.
She would have, of course, opted for IDCT in the cervical area. So, and this is the thing, and Kevin and I and Bob have talked about this numerous times about, where Kevin says, where I'm doing a multi-level fusion, could I possibly save that adjacent level that's on the bubble to be fused or not to be fused? Could we do that? Or one of the things Kevin's discussed in great detail is there a possibility once he does a discectomy that we could possibly, get that annulus closed properly and then inject from the contralateral side to basically restore some function to that disc after discectomy. So I don't know, Kevin, you might want to talk about that briefly too, because that's another huge area of patients that have some issues after discectomy.
Yeah, well, so one of the things any spine surgeon will tell you guys is patients will frequently ask, well, doc, what are you going to put back in there? And of course, you have to say, well, nothing. So sure, it would be nice to have something that worked in terms of potentially improving the discs that have already herniated and trying to prevent future problems at those levels.
Yeah. And quite frankly, to expound on this a little further, these would be supplemental BLAs. Once we have, and we've shown such great safety, we believe the FDA would be very open to these supplemental BLAs of adjacent level injection after or during fusion, possibly this whole notion of injection after discectomy along the way. So, and we think through that supplemental BLA with the comfort that we've established around the safety and hopefully the success of our pivotal phase 3, we can really begin to move the ball forward and accelerate treatment to all these patients up and down their spine.
The other part of it too is that this cell in the manufacturing process will be approved with the lumbar indication. So it's not like we have to go and come up with a different cell derivative or different manufacturing. It's a different volume, and the delivery technique, different needle the practitioners will use will be the differentiator. So, we'll get some light speed, with this approval to work on some of those supplemental indications.
Do you have some preliminary revenue forecasts?
We do. After commercialization, we believe by year 3 we'll be tracking revenue to $1 billion, to a blockbuster status. That's a blockbuster drug is known as a drug that does $1 billion in revenue annually. We think that that by year 3 we'll be ramping to that $1 billion revenue possibility going forward. If we do see and get the labeling that we believe the phase 1/2 data suggest in the high dose patient.
And Flag, I get the sensation I feel when I'm talking to you.
Yes, sir.
You're talking about your wife.
Yeah.
I see the mission. I see it in your heart.
Listen, yeah. And go ahead.
No, I want to tell you the founders that we've seen successful are the ones that have that mission.
Well,
and I want to,
yeah. And look, for me, it was different because I was in the surgical, and for Kevin, of course, and Bob too. I mean, Bob and we refer to ourselves as former metalheads. I had a surgical specialty distribution business, and we sold plate, rods, and screws and fusion biologics. And I would walk out of these surgeries with a $15,000 to $25,000 PO around plates, rods, and screws, really with my emphasis around helping the surgeon with the surgical microscope. And we bundled kind of off the surgical microscope used in neurosurgery, of which, was a key piece of Kevin in developing this whole notion of minimally invasive spine surgical techniques utilizing the operating microscope. And that's where Kevin and I, sort of found each other in 1991. And I would walk out with this order, or Kevin or somebody would be using the surgical microscope to do a microdiscectomy, but if the joint was unstable, he'd push the microscope out of the room, grab a scalpel before Kevin's techniques became available, make an 8-inch incision, and sort of commit this, a very controlled amount of barbarously by putting plates, rods, and screws in these patients. And I would walk out in my inner dialogue with going, God, I'm grateful for the business, but there's got to be a better way. And so my wife had this cervical fusion in 2004. And, we knew that adjacent level disease would happen over time. And, so I found this technology. Kevin had been aware of it before me, but this technology came to the forefront through a call from Dr. John Robertson, who was the chairman at the University of Tennessee. And he said, take a look at this. This is amazing. And all kinds of light bulbs went off. And, I'd been in the surgical device distribution business for a long time. I was a little bored. And I really felt led that this could be a way that I could help millions of people. And of course, I was a distributor for Osteotech, where Bob had been the president. And I went back to Kevin and Bob and said, come on, guys, could this really be the answer? Could this really be the grail of degenerative disc disease? And Kevin said, it could, but it's a long walk up a pretty big hill.
So we believed, we had enough faith in this. And I think, I got involved because I certainly had known Kevin over the years and faith in him and in Flag's prowess. We said, it's worth taking the risk and putting the time in and partnering with our loyal investors and building up a team to go after this based upon what the prospect of what this was, particularly with us all having been in the fusion, the metal part of, musculoskeletal for years, and saying, hey, wouldn't this be great if we can, really make this happen and treat patients in a far better way? So it was worth us, committing the time to it and being involved. And it's a long pathway. It's those familiar with device, it's a 510K. This started really in earnest in 2007. So for a lot of us here, it's been a journey, but it's been one of passion and love for what we can do prospectively to patients, like we've been talking about.
Yeah.
And I'll say something else, Jeff, and the graphs don't quite bring this out, but the amount of change in these very symptomatic patients in the trial was impressive. And one of the things I said to Flag, and when we were first looking at the 2-year data, is, my God, these patients are doing as well as someone who has a really well done fusion and does really, really well. In other words, It's a darn shot, and you're seeing outcomes that are remarkable.
Yeah. I mean, Jeff, if you notice that durability graph that shows the durability out to 2 years, you can see from a Oswestry disability, they're patients that are in the 3rd and 4th quintile of disability that we're driving back. As you see over time, at the end of 2 years, we're back to the 1st quintile of disability, which is pretty much no disability at all. It's absolutely incredible. And, it again, yes, it was a smaller patient population in terms of how we did, the study, but it was still a randomized, double-blind placebo vehicle controlled trial. And the data, especially compared, while we couldn't compare between groups, we could compare within groups with MCID. And you can see we did extremely well in multiples of MCID relative to the other 3 arms. So, it's very encouraging data. We're very excited. The FDA was certainly excited in that they granted a regenerative medicine advanced therapy designation to us. And they really are helping, and they are very enthusiastic, but they will not hedge one iota on the rigor, and that's up to us, and we get it. And we believe that we've put together a very good pivotal phase 3 plan for the FDA, and we're looking forward to continuing our discussions there. We've from our CMC, which is our chemistry, manufacturing, and controls. That's all of our quality around our manufacturing technique and the scale-up ability in our built-for-purpose facility. We've got a great pathway that's already signed off by the FDA. So we feel very good about that. We just need to make sure that we're applying the same rigor in the phase 3 clinical trial moving forward.
Talking with multiple founders of companies, I've learned that a red flag is when they continually talk about an exit or an exit strategy, right?
Yeah.
And when I talk to each of you about what you're trying to do, the overwhelming mission is to make lives better.
Yep. Period.
And not only is that just refreshing to hear from a clinical care or just a human perspective, that ultimately becomes the founders that drive the success. So it's very refreshing to hear. And I wanted to ask, Bob touched on, how are you going to get this to the market? We answered it in the deep dive, but in a succinct manner, what's the marketing strategy here, Bob?
So we spent time putting together a commercialization plan. I think a lot of it ties right into our pivotal study strategy, is that the heart of treating patients is partnering with the appropriate treatment centers out there. So that's the heart of it. So ones that are integrated spine practices that tie in the surgical plus the interventional, where we can channel in patients where they're going to be appropriately screened and injected per what the labeling of the product will be. This will be tied into, careful selection in each market area with pharma-based advertising, where we're going to, create awareness for the patients and be able to channel them into the treatment centers, working closely with the payers and all the different tiers of payment across the market to, get this therapy approved so we can treat this vast universe of patients who are suffering from this. So that's the big part of it. Flag touched on this, and Kevin too, the rigors of doing an appropriate pivotal trial. So, we can confirm the safety, the efficacy for reduction in pain, disability. And, a big thing with the FDA and us wanting to do this right is that prospect of restorative. We touched on that there aren't other things in this mild to moderate space that are restorative that can actually, arrest the disease state progression going on. So, we're optimistic that we'll be able to confirm that in this pivotal study, and we're certainly going to market that within our labeling to the fullest to the patients to deliver something special to them.
And as a follow-up to that, Bob, is your team familiar with the American Society of Interventional Pain Physicians?
We are.
Kevin, yes.
Yes.
So they hold an annual meeting in January, typically in Las Vegas. I think they alternate with Orlando occasionally. That would be definitely a place that we should bring this to once this is ready for commercialization.
Fantastic. We'd love to, and we'd love to come next year. And depending on Kevin's schedule, I'm always available. Bob's available. We could present there, and we certainly have some clinicians from our phase 1/2 trial who would be glad to present as well. So, we would welcome that opportunity.
I know there are factors out of your control, but what's your best guess on going to market?
So we're very hopeful in the U.S. that we can go to market within 3 years. We're going to have to do a very good job on our phase 3 trial to get through that. We are looking at this whole possibility of a conditional marketing approval in Japan, and we are looking at other opportunities outside the U.S. where we could set up a patient registry where the patient could get a dose and as part of that registry. So please stay tuned. We're excited about our progress and where we are. We are progressing in a very careful and very conservative state to do the right things to meet that rigor, but also to try to get this to as many patients as possible as quickly as we can, especially being buoyed by the fact that we've seen such great safety in both the U.S. and the Japan trial. We feel very comfortable giving this to patients, and I certainly feel very comfortable in giving this to anybody in my family.
Tell me about the funding round you're in now, the capital needs, current valuation, pre-money, post-money, and what you said it's convertible note. At what point does that convert? At what event, et cetera?
So we want to raise about $45 to $50 million in the convertible note. We've got about 30 committed. We're looking to, and this note has a 20% discount. It has an 8% coupon. It all converts to preferred shares in the company at the price that we negotiate with the lead funder in the Series D. We do have a value cap of $620 million on if the company were to be bought within the time frame of the convertible note raise to the time that we negotiate the final price with the leader in the Series D. I think you never know about the possibilities of where that is at. We do have many both device infusion medical device companies following us and big pharma, and we have very good relations with all of those folks, and they're watching us very carefully. But by the same token, we're basically in it to get this to millions of patients, knowing full well this is a tremendous standalone business by itself that could be developed into an Edwards Life Science. We are looking at liquidity really around doing an IPO, then followed by maybe keeping it private. And obviously M&A is in there. I never thought I'd sell my surgical device distribution business, but Itochu, one of the 5 trading companies of Japan, came along and offered me a lot of money to sell that business to them. And I felt like I could do better, and I had this sort of intuition of another calling, and lo and behold, 3 years later into my 5-year earnout with Itochu, John Robertson calls me on the phone, and I reach out to Kevin and Bob, and we start this business. And of course Itochu gave me their good graces to follow this as I finish my earnout. So it's really interesting and fascinating, and Providence has been alive and well. So we're going to continue to do exactly what we need to do by staying white hot focused on the execution of IDCT first, to get this to patients with having a good commercialization plan ready to go, and then these other projects around our preclinical development in this next cell that produces more collagen, and then other things as we move forward, and then filling out how we can serve the patient up and down the spine from a degenerative disc disease standpoint. With that, I'll hand it over to Dr. Foley. This is a recent presentation he had made. I'll let Kevin tell you a little bit about this and then go through the deck. Dr. Foley, take it away. Thank you.
This is a presentation I gave just a couple of weeks ago down in Paradise Island. There is an annual regenerative medicine summit that's held there. called HEAL-INC. And we were there to present our data, and I'll summarize today for the audience. These are my disclosures, and I do serve as the Chief Medical Officer for DiscGenics. Symptomatic lumbar degenerative disc disease is a common problem. Affects millions of people, not only in the US, but throughout the world. And it's estimated that just within the US, we spend over $100 billion annually treating this entity. The current treatments for patients with symptomatic DDD that does not respond to typical non-operative management are limited, and none of them result in repair or regeneration of the disc. And down at the bottom of the slide, I've got the classic Furman scale for the grading of the degree of disc degeneration, and we're targeting here Furman's grade 2 through 7. So when you have a patient who presents with predominant low back pain, classical mechanical low back pain, who has failed to respond to typical non-operative intervention, exercise programs, non-steroidal anti-inflammatories, activity alteration, chiropractic care, injections, etc. The options are limited for those patients. They consider a lumbar fusion, but we know for back pain alone, patients who don't have predominant leg pain, fusions are not ideal. There are mechanical replacements for the lumbar disc. They've been around now for well over a decade, and they just haven't worked out that well. That's why they're not nearly as accepted in the lumbar spine as they have been for the cervical spine. And so ideally, in treating these patients, we'd get to the biologic basis of the disease and restore the disc structurally and functionally. So our discs are shock absorbers. In fact, they are hydraulic shock absorbers. The center of your disc, the shock absorbing portion, is called the nucleus pulposus, and it's almost 80% water. So much more water content, for example, than cartilage. Cartilage is about 50% water. And this hydration is key to the function of that disc. Within your nucleus, in the center of that disc, there is a sparse population of cells called nucleus pulposus cells. And these cells make the matrix, the extracellular matrix, or ECM, that makes up your nucleus. And one of the key components in that extracellular matrix is proteoglycan. The proteoglycans have negative charges, and they attract positively charged ions like sodium. And this is what sets up the osmotic gradient that lets your disc stay hydrated. The collagen 2 that sits within the nucleus helps corral or enclose these proteoglycans, and it's also important to the disc maintaining its hydration. So as the disc loses its ability to make proteoglycans and to make collagen 2, it loses its ability to hold on to water, and it loses its hydraulics. So again, you have predominantly collagen 1 to give you your structure to the annulus, the outer stiff container on the disc that holds the nucleus in, and then the collagen 2, as I mentioned, functions to help the nucleus maintain its hydration. So within this nucleus, within your nucleus pulposus in the center of your disc, there's an active metabolic process going on. And so your nucleus pulposus cells are constantly making elements of the extracellular matrix, or ECM. And then there are enzymes in your disc, such as MMPs, or matrix metalloproteinases, that are degrading the ECM. And there is a balance, a healthy balance, between the production of the ECM molecules and the destruction of those molecules, a homeostasis. In disc degeneration, that homeostatic balance becomes upset, and the destruction of your extracellular matrix starts to outpace your body's ability to make new ECM. And as that ECM degrades, your disc darkens on an MRI, it dehydrates, it loses height, it becomes stiffer, and it just becomes a less effective shock absorber, and it becomes prone to damage from just normal daily activities. So in looking at targeting a disc biologically and looking at a biologic answer to this disease rather than placing an artificial device or bone in your disc and fusing it, one ideal target would be the architecture of that extracellular matrix. We've just discussed how that architecture is degraded in typical disc degeneration. And so restoring that architecture is a logical biologic target. The other phenomenon that happens as you get that homeostatic imbalance and you start to degrade that extracellular matrix, as those normal molecules fragment, the microfragments actually are foreign bodies within your disc, and they elicit an immune response and an inflammatory response. And so we know that a number of inflammatory cytokines are upregulated in disc degeneration. Tumor necrosis factor alpha, or TNF alpha, actually having the largest presence, but all the inflammatory cytokines I list here actually get upregulated when a disc degenerates. Here's a typical profile of cytokines in a normal disc or healthy disc in the light gray, and then a degenerated disc in the dark gray. And you can see how those inflammatory cytokines indeed are upregulated when a disc degenerates. So this immune response leads to inflammation, and there's a reactive innervation of that disc. And so that innervation is pathologic. Those nerves are not supposed to be there, and they become inflamed too. And that's thought to underlie the genesis of the pain in a degenerated disc versus what goes on in a healthy disc. So another logical biologic target beyond restoring the extracellular matrix is to directly attack that inflammation. And that's probably why, for example, Jeff, that anti-inflammatories can have some impact in symptomatic DDD, and corticosteroids can have some impact, but we need something more potent. So one approach to attacking both of these biologic targets, to restoring the extracellular matrix and mitigating the inflammation, is to provide a new functional cell population in that disc. And with those functional cells, you can actually address both of those targets. You can provide a platform to actually make new ECM. And then you can also produce paracrine signals that can modulate the inflammation. And this, in fact, is not a new notion. In fact, a number of different cell populations have been investigated for this potential. And so for years, investigators have tried to find a cell population that might achieve these goals that we just discussed. And I list them all here. And to date, prior to the evolution of the cells we're going to talk about today, there just really hasn't been great success in this space, despite the use of a number of different cell types in this quest. And it was based on the logical nature of this approach, but the lack of effectiveness of other cell populations that our group decided to explore the possibility of a unique cell population for this problem. And so these cells we term Discogenic cells. And these cells were first derived in the lab at the University of Tennessee and within the Department of Neurosurgery. They're an outgrowth of culture techniques that were used to derive progenitor cells, stem-like cells from the adult human brain. And those techniques applied and modified for the disc allowed for the development of a new progenitor cell population that we term Discogenic cells. So they're derived from adult human disc tissue. We manipulate them in the lab through a series of culture sequences that that change the cells phenotypically, don't change the genotype, as we'll get into. And then we can cryopreserve these cells, and they're mixed so they can be stored long term. And then they can be thawed at the treatment site, mixed with a viscous carrier and injected into the patient. So these cells are what is termed pluripotent. They can produce other target tissues depending on what we add to the culture media. So as we derive them, they naturally are driven to become mature nucleus pulposus cells. But we can take these same cells, we can drive them to bone, we can drive them to fat, or we can even drive them to cartilage. So we've spent years characterizing these cells, years in the animal lab. And so we know that as we put these cells through the culture system, we change them phenotypically. They change in shape, they change in terms of their cell surface markers. And some of those changes actually prove quite helpful for our use. For example, there's a significant downregulation of the HLA, DR, DP, DQ cell surface markers. Those are the cell surface markers, by the way, that underlie organ transplant rejection. And so if you can downregulate HLA, DR, DP, DQ, you can produce a cell that's immune privileged. In other words, doesn't incite an immune response even in a foreign host. And then in culture, after we've derived these discogenic cells, they start to produce the constituents in culture of normal ECM. And so they produce aggrecan and the other proteoglycans that are in an adult disc. They produce collagen 2, necessary to corral that proteoglycan population. And they produce some of the other constituents in the ECM, such as what's called an actin cytoskeleton. Speaking about the properties of these cells, there are ways to look at cells to see whether they are inflammatory or immunogenic by pitting them against a foreign white cell population, typically lymphocytes. So in this particular graph, when you pit human peripheral white cells, mononuclear cells, and add discogenic cells to them, you very much downregulate the cell's response to inflammatory stimuli. You can also mitigate how cells react to each other. This is a so-called two-way mixed lymphocyte reaction. And the blue bars are what happens if you mix two unrelated white cell populations. They essentially attack each other and they multiply. And then you can see the mitigation of that reaction if you put the two cell populations against each other in culture and then simply add the discogenic cells. Significant improvement or mitigation of that response. So we know these cells are immunomodulatory and we know they're anti-inflammatory. It's also important when you're developing a cell population for human use that you show that you're not going to be forming tumors. We all know that cancer cells are essentially, immortal stem cells, right? And you don't want to inadvertently introduce a population of cells like that in humans. And so there are ways to test the tumor forming potential of various cell populations. We know in our cell population that not only are they karyotypically normal when we look at their chromosomes, but they don't form tumors in vitro, and they don't form tumors in vivo. So one of the classic models looking for tumor formation in a living organism is the athymic mouse model. And you typically test your cells against a known tumor forming cell population. So the typical control population are the HeLa cells, the Helen Lane cells that form cervical cancer. So if you place HeLa cells subcutaneously in the athymic mouse, you get cancer everywhere in diffuse fashion. When we place Discogenic cells subcutaneously in athymic mice, not only do we not see any tumor formation, we actually see bits of disc form subcutaneously in the mice. And this is interesting. These are the human cells, by the way, in the mice. So over the years, we've done numerous animal studies in numerous species: mice, rats, rabbits, pigs, even dogs. In all these studies, we've used the human cells, knowing that it's not only a way to test the cells in terms of their ability to treat experimental DDD, to look at the histology, to look at the chemistry, but also to test whether or not there are going to be adverse events, and especially in immune response, because in the animals, of course, the human cells are xenograft. They're not allograft. They're worse than that. They're foreign. So in these studies, we are inducing an experimental model that mimics disc degeneration, and the way to do that classically is to stab the disc with a needle. And if you take a rabbit, a New Zealand white rabbit, for example, and on day 0, simply puncture the disc, nothing more than that. Then by 2 and 4 weeks, you can see the bar graph or the linear graph on the bottom. The discs diminish significantly in height, and then that height doesn't return. So the yellow line doesn't show up as well on this. It's where the asterisks are, is a cell population where we've injured the disc, but then at week 2 added our cells. And our cells, instead of the disc continuing to diminish in volume, consistently in all the animal studies, the disc volume increases. And not only does the disc volume increase, but the histology improves. So if we puncture a healthy disc, not only can we see the disc height diminish radiographically, we can see the height diminish histologically. And then at 2 weeks, if we take that injured disc and then inject our cells, we can watch that disc volume improve, as I just said. See it not only radiographically but histologically, but we can also watch the cells improve morphologically. So on the magnified view and on the top left, next to the healthy disc, is normal nucleus pulposus. The cells are plump. There's ample extracellular matrix, ample cytoplasm within the cells, relatively plump nuclei. When you stab the disc with a needle, not only does the disc diminish in volume, but the ECM contracts, condenses, and the nuclei shrink. They become pycnotic, as you can see. And then when we inject our cells 2 weeks after having stabbed the disc, we restore histologically something that looks more like a normal disc histologically. We see the ECM increase. We see the disc, the nucleus pulposus nuclei plump up, their cytoplasm increase to a more normal state. And once again, just this slide is to emphasize that through the culture system, we downregulate the surface markers that are associated with activating an immune response in the recipient. And so we've seen in all of our animal studies no signs at all of any rejection. So based on this abundance of laboratory data, basic in vitro data, and then the in vivo multiple animal studies, we petitioned FDA to carry out a first-in-man trial. And that was the multi-center trial that we recently completed. Involved human subjects enrolled in 14 different centers across 12 different states. And subjects were screened, of course, for inclusion and exclusion criteria. And if they met those criteria, then they were allowed into the study. And basically, these were subjects between 18 and 75 years of age who had a diagnosis of early to moderate DDD, modified Fuhrman grade 3 to 7 on an MRI, as we talked about Fuhrman grades a little earlier in this talk. They had to have had chronic low back pain for at least 6 months prior to their screening visit. Most of them had it for much longer. They had to have been unresponsive to at least 3 months of non-operative management. Again, most had much more than 3 months of non-operative management. And they had to have a certain amount of low back pain on their visual analog scale at screening, and a certain amount of disability on their Oswestry Disability Index. And the exclusion criteria were really geared towards trying to exclude patients who had other causes of their low back pain rather than degenerative disc disease alone. And they couldn't, for example, have a symptomatic radiculopathy due to nerve recompression. So these were patients who had chronic axial low back pain, recalcitrant to a trial of non-operative management that, as best we could tell, were symptomatic from their DDD. And the tiebreaker in patients who had more than one level of degeneration on their MRI was discography. So if the subjects met the entry criteria, they all got a single 1 cc injection, and they were randomized to one of 4 treatment groups. They either got a cc of carrier mixed with 3 million cells, or they got a cc of carrier mixed with 9 million cells, or they got a cc of the carrier alone, which was sodium hyaluronate, or they got a cc of saline. And we based the cell dosing, so this was a dose escalation study too. We based the dosing on what we had seen in the animal discs, and we simply scaled for human disc volume. So we knew the cell dosing that was working in the animals, and simply upped that dose based on animal disc volume versus typical human disc volume. So the subjects were blinded to treatment, and they were assessed by blinded clinicians. We excluded the injectors from being the assessing clinicians, because we knew that even though the injectors weren't told what they were injecting, there's a difference in the viscosity of putting in a cc of saline. I'm sure, Jeff Brown, you know this well, versus putting in a cc of sodium hyaluronate. You'd be able to tell,
Sure.
Right? And so we didn't want any bias on the part of the assessing clinician. So the assessing clinician had to be someone who didn't do the injection. And so the patients were called, it was typical outpatient injection, quick injection, and out they went, done under local sedation protocol as to whatever the center used for its typical protocol. And then the patients were followed at 4, 12, 26, 52, 78, and 104 weeks. And they got clinical exam, they had their patient-reported outcome measures gathered, and then they had imaging, plain films and follow-up MRI. So MRI and plain films at study entry, and then follow-up clinical and radiographic studies. So 60 subjects were enrolled in 14 centers across 12 states, and there were 20 high-dose patients, 20 low-dose cell patients, 10 saline alone, 10 vehicle alone. Most common Fuhrman score was 4. Most commonly observed target disc was L5S1, although you see there were a smattering at the other levels. And in negotiation with FDA at the start of the trial, you have to set an efficacy endpoint. So the efficacy endpoint was set to be at 52 weeks, a statistically significant reduction in pain from baseline to week 52 greater than 30%, as measured on a 100 millimeter VAS scale. And in fact, that was only met at all time points in the high dose cells. And actually the pain relief in the high dose group was over 60%. There was some improvement in the saline group. Saline does have an effect, as other studies have shown, but it only demonstrated statistical significance greater than a 30% reduction at week 26. So here are the pain responses. And black is saline, the blue is the high dose patients. So with an injection of high dose cells, by the 12-week visit, there had been really a profound improvement in pain on 100-point VAS in those patients. And that was sustained out at all time points. It still held true at 2 years. You see that saline briefly at 26 weeks did reach statistical significance, but by 2 years, saline is trailing off. We did see some late response with both the low dose cells and with the carrier, but nothing achieved the consistent results of the high dose cells.
Jeff, we did some post hoc analysis around these pain studies, the pain scores in the low dose and the vehicle. And the low dose does seem to work over the long term. And the vehicle, we noted that more vehicle patients were taking opioids to reduce their pain. So it was very, very interesting, as we did some of this post hoc analysis to see the differences and how well the high dose did from 12 weeks all the way through.
Yeah, the only group that had diminished opioid use was the high dose group. So yeah, there's some confounding of those results because of opioid use by some of the other groups. And what we found that was striking though were the functional results. So the VAS, as you guys know, you're asking the patient, they get a 10 centimeter, 100 millimeter horizontal line, and you say put an X where you're hurting. If you woke up on the wrong side of the bed that day, if you had an argument with your significant other, who knows where that X grows. The functional studies, much more in depth. So Oswestry disability, it's a 10-question test, and with multiple choice answers for each of those 10 questions. So a little bit more thought goes into that. And it's interesting to look at how much change there was in the disability, improvement in disability in the high dose patients as compared to the other groups. So we saw some improvement in all groups, just not the profound improvement that we saw in the high dose group. And this notion of an MCID. So an MCID means a minimum clinically important difference. So in Oswestry, if you have an Oswestry in the high 40s, low 50s, you're disabled. And if you're a normally functioning human being, you're going to have an Oswestry somewhere between 15 and 20. Hardly anyone has an Oswestry of 0. And so a 15-point change in your Oswestry is a pretty typically agreed upon clinically important difference. In other words, something a patient really finds meaningful. And we saw 30 points of improvement in the high dose group in the MCID, twice the agreed upon MCID. And similar with the EQ5D, this is the European Quality of Life 5-dimension survey that really looks at what your quality of life is like. And an MCID and an EQ5D has to be 0.08 to be meaningful. So we almost 3 times the improvement in the MCID in the European Quality of Life survey in these patients in the high dose group. And again, a prompt response by 12 weeks, and prompt and profound, and then out to 2 years still sustained. So these are the aggregate results. So with a single shot, so Jeff, like you're doing someone in your, comes in, you've got your fluoroscope, you prep them, give them a shot. Mm-hmm. 12 weeks later, profound improvement in pain, profound improvement in their level of disability, profound improvement in their quality of life. And again, it's sustained out to 2 years. Importantly, no serious TEAEs, treatment emergent adverse events. So we just didn't see any. And it's the same we've seen through our animal studies. So kind of in keeping with what at least our preclinical data seem to indicate safety-wise, our human data so far demonstrates that the cell population is safe. And the other interesting thing, remember, we're imaging these folks, and the we used an independent imaging core lab, medical metrics out of Houston. So a core lab of assessing radiologists who have nothing to do with the study other than they're reading for us. And they have an automated measure of disc volume, doesn't actually even involve human interaction. And I'll show you that data here in the next slide. So here's the disc volume data. So from baseline, the only group that had a sustained improvement in disc volume was the high dose group. And in fact, it was still increasing at 2 years. You know, we had seen the disc height improvement or disc volume improvement in the animals. So it was very good to see it, of course, in the humans. And interestingly, in the other groups, you know, the more sophisticated, the more far along the phylogenetic spectrum the subject is, the more injury you've got to do to a disc to induce that degenerated type state. But Jeff Brown, you probably know there is some data out there that shows that even injecting a disc for discography can hurt it.
For sure.
And so look what happens, for example, in the saline group in dark.
Right.
You know, their disc volume's going down at 2 years. The carrier in red's going down. The low dose cells in green kind of make a late comeback to baseline at 2 years, but the high dose cell group, consistent improvement in disc volume. In fact, we were so interested in this phenomenon that we're continuing to follow these patients out beyond 2 years, the high dose patients, to see just, you know, not only how they do clinically, but what's going on with disc volume at 3, 4, and 5 years. So more data to come. So the phase 1/2 trial demonstrated that high dose allogeneic disc progenitor cell therapy with Discogenic cells was safe, well tolerated, and it produced clinically meaningful and statistically significant improvements in back pain, function, and quality of life by 12 weeks. And these changes were sustained out to 2 years following a single intradiscal dose. The disc volume increased significantly in the high dose cell therapy at 1 and 2 years. And coupled with our laboratory data, where we're able to look at the histology, right? We haven't been able to look at histology in one of our human patients yet, needless to say. But based on what we've seen histologically in the animals and what we see with the volumetric data and clinical data in the humans, we believe that the increased disc volume in the human trial is consistent with a regenerative effect of our cells.
That is fantastic. I'll summarize it for a layperson. You tell me if I've got this right, but you've got a product that decreases pain, decreases inflammation, increases disc architecture. It's non-immunogenic, so you can't have a allergic, like an organ rejection type process.
Yes.
There's no risk of tumor growth. You've got, and this isn't a claim, this is what we think, both clinically, radiographic, and at least in animals, histological evidence for efficacy for a problem that affects millions of people yearly.
Yeah, we think we have something very interesting here, Jeff.
I mean, sign me up.
And very useful, because as you know, there's just not a good answer for these folks.
There's not, but this may be it.
It's a labor of love to help. You know, it's our opportunity to literally help millions of people. Kevin's already done that, being the father of minimally invasive spine fusion. I think his reach has reached out to that many people, and I think he's going to do it again together with us. We're going to do it together where we literally can help millions of people around the world, and that's really what, you know, I think, you know, the essence of life is to help others, especially when you see something that can be brought to these patients. As you know, Jeff Brown, as well as anybody, all three of the doctors. I mean, you know, y'all take a pledge early on, and we're just trying to maintain and grow that so that we can serve these patients and get them back to good health.
That's fantastic.
Thank you.
I wanted to tell Kevin, you didn't know this, but in the late 90s, I actually worked with Valari.
Really?
I actually cultured a lot of
Didn't
these.
know that.
I did. I
Wow.
did cell culture for him in the late 90s. And
Huh.
Valari was awesome. He smoked a lot. He said he
Oh,
wanted
well,
to
you know, it
protect his brain. He
well, it
protect his
ultimately did him
brain.
in, you know, and you know, he did. I mean, he did some phenomenal work on adenocarcinoma as it metastasized to
Yeah.
the brain. And
Yeah.
this was the original process, is that he was going to grow all these daughter cancer cells that he could do pan-pharma testing against. And it's a similar culture system where we do this selection of cells from the disc tissue and then grow them out.
Mhm.
Amazing.
Well, he was Russian, and I was perplexed that he didn't really drink. He said but he smoked. And I remember they would do tumor resections and bring the tumors up to the hood. And I had a hand in developing your products, you know, guys.
Oh.
Yeah, we're going to close up. We've been on for 2 hours, but Kevin, thanks so much. Rarely do you meet people with the match of humility and clinical competence and skill that you have, brother. You're just great to the world. And you never cease to amaze me how humble you are despite your accomplishments. And I really thank you for your time. I hope that this broadcast will bring you guys the investors that you need to move forward, and also expose the product that you're bringing to potential users of it, and help a lot of people. So thank you for joining us. I'm Jeff Brown with your Merge Medical podcast, and with my host, Dr. Jeff Cole. Thank you for the executive team from DiscGenics for joining us today.